Assalamualaikum. Thank you for the kind introduction. I'm going to time myself so I don't go over time. And I'm also going to check if my slides are working. All right, cool. So I'm going to try to make this interactive by starting with a quick survey. So can you guys feel your one of your earlobes? Is it attached to your ear or is it detached? So maybe like you can shout out a few answers. Huh? Attached. Anybody like dangling like mine? Detached. So how many detached, approximately by raise of hand? All right. And what about how many attached? All right. Fine. All right, cool. So scientists actually went in and like wanted to scientifically answer this question, like what causes the structure of our ear? So what they did was they collected data from thousands of individuals. They literally asked them this exact question, what I asked you, like, is your ear attached or is it dangling? And what they did was they interrogated their genome and tried to find any correlations. So this plot below that you're seeing, this looks very complicated, but it's essentially called a Manhattan plot because some of the like skyscrapers, some of the genes kind of pop up on top. So what they saw was that there were like about 6 to 7 genes which are involved in how the structure of your ear is formed. And in addition to that, essentially is your earlobe attached or is it detached? So what do we learn from this? We learned the fact that through a combination of genes working together, the combination of genes results in what essentially we look like and who we essentially are. All right, so I'm going to ask another question now. Are both of your earlobes the same or are they different? Same. Anybody with different? There's one. That's super rare though. For most of the time, your earlobes will likely be the same. And the reason that they're likely going to be same is because they have the same DNA. They were both given the same blueprint to kind of form into the structure that they are formed. All right, I have another one, and this one's a little more interesting. Do you have a body odor? And I feel free to smell yourself if you want. Or maybe ask the person right next to you. Another way, a nicer way to put it is, do you feel the need to use deodorant or perfume often? Anybody brave enough to answer this? Do you have a body odor? Okay, so clearly nobody in this room has a body odor. And it's entirely possible. So the thing is that body odor is actually pinned down to one gene, a gene called ABCC11, which is expressed in our sweat glands. And what we've seen is that there are people walking around with this gene essentially turned off. And what it means by turned off is like, because that gene is turned off, they don't generate a body odor. A little bit louder. They're having a tough time hearing on the least. No worries. And people actually went in and saw like how common is this off mutation for the ABCC11 gene. And what they saw was that 9% of the people worldwide actually carried like a completely off version of this gene, which means they did not have body odor. For the rest of it's either partially on or it's 100% on. So just by those numbers, like about 9% of the people were actually true. The other ones were just not brave enough to raise their hands, maybe. The other thing to also note is like, it's not randomly, like this off mutation, it's not randomly distributed around the world. There's actually like some level of structure. So in the map below, the red allele, the red version is essentially the off version, and then the blue is the on version. So you can see like in East Asia, it's actually about 75%. South Asia, it's about 45%. And for Europeans and for other populations, it's actually much lower. So we learned two things from this. Number one, that our genes, by affecting our traits, they can also affect our behaviors, like how much perfume we would apply or how much deodorant we would apply. Secondly, that these genetic mutations, they're not randomly distributed, but there's some geographic structure to those things. Now, another thing that we need to kind of address is, does genetics determine everything? So I'm going to ask a few questions over here. So the question is like, do you think this trait is genetic or is it not genetic essentially? So for eye color and skin color, by a quick show of hands, like how many people think it's a genetic trait? All right. So most of the people. What about this next one over here? Educational attainment. How many people think this is genetic? All right, one person. All right. So in actuality, like all of these traits, essentially, they have a genetic component. How strong that genetic component varies from trait to trait. So for example, like for education attainment, there've been like many rigorous studies, and people have actually said that it has a genetic component, but it's less than 3%. 97%, it's about how much you study. Versus for eye color and skin color, essentially, those are highly genetic traits, and those are primarily driven by our genetics. And then there are other traits, like, for example, height, cholesterol levels, which sort of fall between like in the like mid ranges, essentially. So what do we learn from this? That your genes and your environment essentially do create you. And this is a schematic that I like to show a lot, is because this kind of really drives home the point, where you have two plants which are exactly genetically the same, and they're put in uniform lighting. The only thing different is their environment, aka their soil. And so the difference between these two groups is totally environmental, despite their genetics being the same. However, despite the fact that this is a trait which is both determined by genetics as well as the environment. Now, why is studying this important? In my opinion, this actually is studying this important is because Allah actually mentions this in the Holy Quran. And the translation of this verse, this is from Surah Ar-Rum, ayat number 23. And the translation is, and among his signs is the creation of the heavens and the earth, and the diversity of your tongues and colors. In that surely are signs for those who possess knowledge. So what Allah Ta'ala is encouraging us in this verse is to study the diversity within our tongues and within our colors for signs of Allah Ta'ala. Now, for me, the way I kind of look at it, and once again, this is my personal opinion, is especially the aspect of looking at the diversity of tongues and colors. So tongue is something that's essentially a product of your culture. The language you speak is, and the way you speak it is primarily a product of where you were born. And we've seen this, we obviously see this, like, for example, for immigrant parents, their children, the way that they speak, obviously pick up their surroundings. Versus skin color is something that's more innate. You're essentially born with it, i.e. it's determined by our genetics. So obviously, looking at the diversity of languages is a field on its own, and I won't be going too much deep into that. I'll be focusing primarily on the diversity of colors, which is primarily determined by our genetics. So in my view, like when I'm looking at this verse, Allah Ta'ala is saying that look at the diversity of our color, and that color is coming from our diversity of our genetics. So after 1400 years of the revelation of this verse, we now have the tools available to us to be able to study the fundamental block of our innate diversity, which is our DNA. And over the past 2 decades, what have we learned as we've looked at our DNA in depth? Well, first of all, we've understand the function of our genes. We've been able to better diagnose genetic diseases. And most importantly, we've been able to help develop cures for diseases. And for the remainder of this talk, I'll give you concrete examples of how we have achieved this during the past 2 decades since the Human Genome Project took off. And then I'll also touch upon like how Pakistan is essentially like a cheat code to kind of accelerate some of these findings. All right, but before we get into the weeds of that, let's go over the basics real quickly so we're all on the same page. So how do we read the DNA? So essentially the way that we read DNA is we collect either blood or saliva, and our blood and saliva carry our DNA. Half of our DNA is coming from our mothers, the other half is coming from our fathers. So our DNA is divided across 23 sets of chromosomes. Within these 23 sets of chromosomes, we have genes. So for each gene, we have 2 copies, one coming from the mother, one coming from the father, right? Now, how do we read these genes, essentially? We read them through a sequencing machine, and the way that it is read is through A, T, C, and Gs. And then what we do is we look for mutations within the DNA. The way that we look for mutations in the DNA is essentially by comparing it to a reference sequence. And what we do is like we identify where from the sample that we're studying, where does it differentiate? Where does it differentiate from a reference sequence? So for example, like from the example in this I've shown over here, there are two, I've just highlighted two differences from the reference genome. The yellow star above is where one copy of your gene is different from the reference sequence. And in the red stars, both copies of the gene are different from the reference sequence. So the yellow star is essentially what's referred to as a heterozygous mutation. And the latter is the one with two differences is what's referred to as a homozygous mutation. On average, a person would differ from the reference sequence about 3.5 million times. So we carry about 3.5 million genetic variants in our body on average. Now, how do we visualize? Now, one way to visualize these genetic diversity is like through this plot through a linear algebra trick called principal components. I won't go too much into the weeds of this, but essentially each point on this plot is one individual, and their genetic diversity has been condensed into one form. And what you see is what I think is fairly extraordinary is like you can see our genetic data segregating along geographic boundaries. So you can see like East Asians are on one side, Africans on one side, Europeans on one side, and then there's almost like this geographic gradient, like from East Asia, South Asia to Europeans, and then there's I think Middle Eastern ones are shown in this one now. And then from the other side, you have Africans going towards Europeans as well. So underneath our diversity of colors is really our genetic diversity. And studying this genetic diversity is how we've kind of better understood ourselves and the diseases, some of the diseases that affect us. So that's great. We can read DNA and we can actually start seeing that underlying our diversity of colors is our genetic diversity. But how does this translate into the clinic? Well, this translates into the clinic by actually studying what do those 3.5 million variants that we carry, what exactly do they do? Now, for the most part, these 3.5 million variants, they don't really have too much of a very large effect. They have either very small effect or no effect. Things start get interesting when you're looking at like the very extremes of the spectrum, especially a set of variants which are called loss of function variants. So what loss of function variants do is they basically turn off a gene. So in the body odor example that I gave you earlier, that body odor gene, like what caused the gene to be turned off was 2 copies of a loss of function mutation. And anybody who carries 2 copies of a loss of function mutation, those individuals, that gene essentially is knocked out in that individual. So I'll be referring to those as knockouts. And one of the easiest ways for us to understand gene function is to simply ask the question, if I turn off a gene, what changes happen? And if we have large enough numbers of people, comparing people who carry 2 copies of a mutation and compare those individuals with the rest of the population, we can actually start to answer this question. And when people figured this out, there was like a mad rush to kind of find as many human knockouts as possible and try to understand as many genes as possible. The poster child of this is like a gene called PCSK9, which when turned off actually lowers your cholesterol levels. And this really brought genomics in towards the clinic where like people were like, oh, you can really tweak your genetics and get clinically meaningful outcomes. But as this mad dash to kind of find out more and more knockouts occurred, what people found out was that finding knockouts is fairly rare. And it's fairly rare and it's fairly rare because the chances of seeing 2 of the same mutations in 2 individuals and then they passing them on to their offspring is fairly low. So if you look at the pedigree on the right, so there's one, the black is let's say a loss of function mutation. The white is like the reference mutation. So if somebody marries obviously a non-carrier, the children would be heterozygotes. Because of this reason, because of the rarity of this, the chances of finding knockouts is fairly low. But it turns out you can double your chances of seeing knockouts. How does that happen? That happens when there's a population with lots of cousin marriages happening. So you can see, like, for example, like there was one mutation in the grandparents, but by the third generation, we actually see one homozygous, essentially a knockout for a particular gene. And the reason that we see it is because on average, two people, the amount of DNA they share is about 2 to 3%, versus the amount of DNA two first cousins would share is about 12.5%. So the chances of seeing a homozygous individual increases significantly when you are looking at a population with lots of cousin marriages. So now if we have a population which has lots of cousin marriages, we can do a lot. So do we know any population which does that? Well, if you look at this map, the darkest point is Pakistan. Pakistan has the highest rates of consanguinity. Consanguinity basically refers to first cousin unions. And I got the opportunity to work in a lab which was DNA sequencing 76,000 Pakistanis. And that is exactly what we saw. What we saw was that, for example, like if you're looking at other places like in Europe or other places in the world, there are about 6% of the genes which are completely knocked out. Versus when we're looking at that number in Pakistan, that number is actually 27%. So the number of genes we can study, the number of genes in which we can ask the question, if I turn this gene off, what happens? That number is 6 times higher in Pakistan as compared to any other part of the world. Now, I would like to kind of want to emphasize this point that having a loss of function mutation does not mean that you're less healthy. In fact, the two cases that I will highlight are actually protective cases where having a loss of function mutation actually gives you a benefit. So loss of function. So I just want to kind of clarify this point that having a loss of function mutation does not mean that you're genetically weaker, or it does not mean that you're more susceptible to disease. It really depends on what sort of mutation and in which sets of genes. I'm going to, really quick over this. So the other reason also that Pakistan is a little interesting to look at is because in addition to consanguinity, you will have a lot of diversity as well. So this is a very similar plot to what I showed earlier, very focused on Pakistan. So you can see in the blue, there's Punjabis. In the red, there's Pathans, and then Urdu speaking, and then you have Sindhis, and then you have Baloch. So you can see the spectrum where it's kind of Punjabis are clustering on the lower end, and then as you go up, there you can see the Sindhis, there are Pathans, and then on the very end, you have Balochis. So once again, you see that geographic diversity, and underlying that geographic diversity, we have our genetic diversity, right? So now I'll highlight two interesting case studies from Pakistan of people who kind of had superpowers because of having 2 copies of a loss of function mutation. So number one, the boy who felt no pain. So in a small village, what researchers identified was like there was a group of people who just did not feel pain. And the reason that they identified this person was there was a street performer kid who used to walk across burning coals. He would take swords and he would just cut himself with it, and he would not feel pain. And when researchers found out about it, they were absolutely astounded that why is this person not feeling pain? And this person's relatives also had sort of these people. And the other thing, what they also noticed was like within this family, there were a lot of early deaths. And a lot of those early deaths were happening because people didn't feel like their children didn't feel pain, and they would like kind of try to jump off something or like completely like try to cut themselves a bit too much, resulting in lots of injuries. It turned out that these individuals had 2 off mutations, 2 loss of function mutations in a gene called SCN9A. And not only that, like by completely turning off that gene, they were not able to feel pain. And this is something that scientists have used in the clinic as well. How many people over here have used lidocaine patches? Right? Most of you. So what lidocaine patches are actually doing is they're trying to mimic this exact mutation. What they're trying to do is modulate this particular gene, this particular receptor, and try to suppress it a little bit so that they can help temporarily numb your pain. So this is an example of how like a finding in humans was able to supplement an actual clinical application. I'll give you another example. So across the world, there's like people are trying to figure out how to lower the disease risk for cardiovascular diseases. One way to do that is by lowering fat. And scientists have known that there's this gene called ApoC3, which helps you lower fat. So within Karachi, like the group that I'm working with, we identified people who had that gene completely turned off. And this is a village near Karachi, and like the green bars over here, like it's a very small community, and it's like lots and lots of cousin marriages going on. And what they did was they went into the village and they basically challenged those people to a very fatty meal. So they gave them milkshakes and they gave them burgers, and then they measured their blood lipids. And in the red, what you see are the people who have that gene completely turned off, and the brown is regular people who don't have the gene turned off. And what they saw was that their digestion of fat in people who had that gene completely turned off was much, much higher, which meant that they could eat more and the amount of lipids in their body would quickly disappear, very quickly disappear. This kind of informed researchers that we can actually knock out the gene ApoC3 and try to lower triglyceride levels, especially people who are at risk for cardiovascular diseases. And now people are actually working on generating therapies targeting this particular gene. And knowing this information that if you target ApoC3 and completely turn it off, it's not completely bad. Like it doesn't have too many side effects. It's very valuable from a pharmaceutical point of view. All right. So I'm almost out of time. In fact, I'm a little over time, but as this like field of genetics is kind of moving forward, I just want to leave you with some of the bold predictions the NHGRI, which is the genomics wing of NIH, is having. The one's interesting is like by 2030, we would know the role of all human genes. Genomics will be used in everyday medicine, and genomics will reveal cures to many genetic diseases. And we're seeing ourselves kind of move in this direction very rapidly. Some things which I find are more interesting, like, for example, hands-on genomic research will begin in elementary school. And also number 8, which I find very interesting, is genome sequencing data will be stored and accessed on our smartphone. So we can just quickly look up and like, oh, we have this mutation. Oh, we have this mutation. We can actually kind of, I guess that can be like a conversation starter whenever you meet someone. So Jazakallah for that. And with that, thank you.